Oh, hello and welcome back for the second part of our symposium. I sense that there are many out there following us. I can see about 113 people who have signed up on the chat, and these are people who can actually ask questions. For those of you who may not know how to do that, on the top right of the page, you will see something saying type a question. If you click on that, it will say join with a guest name. You can just type your name, and then we will be able to see what you're typing and any questions you may have to ask. We're moving into the second part, and we have another very important figures from the interoceptive field. Our first speaker is Professor Catherine Talon-Baudry, who's a senior researcher at the CNRS, École Normale Supérieure in Paris. And of course, Catherine has done very influential work on vision, but over the last decade or so, she has been doing some very influential work on interoception. First, looking at the effect of the heartbeat evoked potentials on perception, then moving on to understanding how this brain-viscera dialogue influences selfhood and subjective experience, and more recently looking at the guts. And we very much look forward to your talk, Catherine. Thank you very much for joining us.
Thank you, Manos, for the invitation. And well, thank you all for being there. So I guess I'm now back to this weird situation where I actually have to imagine you being there. And then I don't get my slides. Okay, yeah, that's fine. Yeah. So Manos gave a very precise summary, actually, of what I'm going to talk about. But the starting point of the story is not really the brain-viscera dialogue. I kind of came to it by thinking about consciousness and what it means to be conscious. And that's certainly not an easy thing to try to define consciousness. And for a long time in cognitive neuroscience, consciousness has been mostly defined by the skills and abilities we have when we are conscious. And that's essentially all high-level cognitive functions like logical reasoning, controlling attention, planning, using language, etc. And in this view, consciousness was this kind of super cognitive function that essentially helps me doing things, but it's a function. And for all kinds of reasons that I will not detail here, it appears that this view has been very useful to get started, but it's a bit stuck into explaining other aspects of consciousness. And just this simple aspect of having a subjective experience is not easily explained by those high-level processes. So another aspect of consciousness that is really central is the fact that what defines consciousness is our ability to experience things. Okay, so when we think of experiences, we tend to think about the kind of cool, fun stuff we want to do during vacation, like base jumping, or if you are afraid of base jumping like me, you might rather prefer to have some nice pâtisserie from a French famous chef, Pierre Hermé, or to enjoy beautiful pictures by Niels Udo. But having an experience is also something that has no particular emotional valence, and that is not particularly arousing, like simply viewing pigeons in a street, or the standard paradigm that is used mostly in vision, seeing a gabble patch. There's nothing particularly exciting about this, but still, the fact that I'm seeing this gabble patch consciously is doing something to me, and that's what we need to explain. So, to try to go in that direction, which is not easy because there's no real path to understand that, to understand why something is doing something to me, it means that it requires a subject of experience. So we can now reframe the question as, how can we build an experiencing subject? So before I dive into what is the hypothesis we are trying to set up and test in my group, I just want to emphasize that this is not a trivial question. That we need an experiencing subject. You might think that because if ever you have a conscious experience, it's happening in your brain. So of course, it's your experience. There's no need for a specific mechanism. But And that's perfectly valid. But if you think about all the unconscious processing that's going on in the brain, now we understand that we need a mechanism to understand why some processing gives rise to this conscious experience, where other type of processes don't. So there is a long-standing tradition thinking that bodily signals have something to do with the self, because bodily signals are intrinsically private. And because I'll be talking mostly about the heart and the stomach, I mean, there's no way for you to experience, for your brain to feel my heart, because you just don't have the right pathways. So we can share things in vision, because we kind of assume that we have more or less the same visual system. But for interoceptive signals, they are truly intrinsically private. So in that sense, they might be self-specifying for the brain. And here I'm talking about a very, very low level of self, like this minimal form of self that would have derived from protective behavior. So that's the self, the organism you want to protect from aggressions. And it's also the self you want to regulate through homeostatic or allostatic regulations. And in this context, the heart and the GI tract are particularly interesting because both the heart, so this you're familiar with it, it's an organ which is life-sustaining, just as the gastrointestinal tract. But both organs are sending constantly signals to the brain. So you're familiar with the fact that the heart is beating. There is an analogous thing, not exactly the same thing, I will detail that later, going on in the gastrointestinal tract. So what could those signals do? They could help building a subject-centered reference frame. And this subject-centered reference frame would enable a central component of consciousness, which is essential for the unity of consciousness, which is our own first-person perspective. And why we need to do something like this, it's because in the brain, information is encoded in very different reference frames. So visual inputs are encoded in gaze-dependent coordinates. You also have head-dependent coordinates. You have somatosensory coordinates. You also have more conceptual coordinates for categories and concepts. And all those coordinates need to be combined at some point, because what we experience is a combination of all this. And there's no region where all this converge. So we need to invent, well, to define what could be a mechanism to combine those information, not only to transform from one coordinate to the other, which is something you might do if you're catching a ball, for instance, but really to hold this together so that you can really appreciate your experience. And it means that this first-person perspective is something which is intrinsic to many, many aspects of our conscious life, from perception and cognition to explicit self-processes and also emotions. So it's great to have cool hypotheses. It's even better when you have tools to probe them. So I guess you're all familiar with what is a visual evoked response. Essentially, you present an image, here a donkey, to a participant. You record brain activity. This is what you see on the right with some noisy traces. And because you don't see much in one single trial, you repeat the trial. So you have more data again and again. You still don't see much. But if you average miracle, you can see that there was some transient that was present in each single trial. So the idea here is just to replace the external stimulus, so the donkey, by an internal stimulus, which would be a cardiac contraction indexed by a peak in the electrocardiogram. So this is a naturally occurring stimulus. You can't control it, but you can still record brain activity in response to this stimulus. So the heart beats normally, so the stimulus repeats, so you can do the same and get a response. Okay, so those are all simulated data, but this heartbeat evoked response that was initially observed in the lab of Chandri in the 80s has been observed intracranially. So for instance, here in recordings by Kern and collaborators in ECOG over somatosensory cortex, or in our own data with an electrode inside somatosensory cortex. So this is a real phenomenon. This said, it's a tricky one when you are using, as we do, MEG or EEG scalp recordings, but I won't get into those details. So we are recording this mostly with MEG. Before I get into the data themselves, I just want to stress again, so what we do, we have MEG data that you would see here in two different conditions, in green and blue, that we get from responses to heartbeats. And this is really what I'm going to talk about, how this response to heartbeats is modulated in different conditions. This is different from what you've heard about and Sarah Garfinkel, for instance, did very interesting work in that respect, which is that if you present an external stimulus at different moments in the cardiac cycle, then this stimulus might be processed differently. Okay, so this is a different story, and at the moment we don't really know how the two interact, but I will be talking about what's on the top line, those green and blue responses to heartbeats. So the first experiment we did, because I'm coming from vision, as Manu said in the introduction, was the simplest experiment ever to study visual consciousness, where participants were looking at a black fixation. When they were fixating properly, it turned red, and then after a variable delay, they would have a faint grading in an annulus around fixation appearing or not. And then after another variable delay, they would get a question mark, and they just would have to say whether they had seen something or not. Okay, so it's a standard vision at threshold. So the gradings were very faint. They could perceive them in only about half of the trials. And what we were interested in was to look at how the brain was responding to heartbeat just before stimulus onset. And what we found is that when participants at the end of the trial would report seeing the stimulus, whether it was a hit or a false alarm, then we could see large responses to heartbeats before stimulus onset. And when they failed to see the stimulus, it was a miss, then we had smaller responses to heartbeats. And this happened in non-visual structures. It happened in ventromedial prefrontal cortex and in the right inferior parietal lobule, so two regions of the default network. So the interpretation we give to this experiment is that for people to say, I've seen the stimulus, it's actually a combination of the experiencing subject indexed by the response to heartbeats and visual information processing. So of course, you should be skeptical. You could think that it's just a global effect. People were, more alert or more aroused or more something very non-specific. But it seems to be not the case. So the first thing is that when we had larger heartbeat evoked responses, it really corresponded to an increase in visual sensitivity. And the second important point is that there was no change, no sign of change in bodily arousal nor in overall cortical excitability. So it seems to be something really specific to the response to heartbeats. But still, this interpretation we are giving is it fits with our hypothesis. It doesn't mean that the hypothesis is correct. So we turned to a different line of research where we investigated more directly the self more explicitly in an interrupted thought paradigm where participants were viewing this gray screen with just a fixation for quite long times. Up to 13 seconds. So it was quite boring. Their mind started drifting. And then at some point, they would be interrupted by those white dots around fixation. And at that point, they would have to rate the self-relevance of the interrupted thought. So if they were thinking, for instance, I was bored, then they are kind of thinking about themselves because they are thinking about how bored they are. So they would rate fairly high. And we were interested to measure heartbeat evoked responses during the thought that was interrupted and later rated, depending on whether the self was rated high or low in the thought. And what we found is that we had larger responses to heartbeats when people rated self-engagement in the thought as high, again in structures of the default network. And here, Mariana Babo-Rebello, who conducted this work, could verify those results in intracranial EEG recordings, where she could, we had patients with electrodes in those regions, so those were two different patients, but then we could correlate the heartbeat evoked response and self-ratings in single trials. And just to give you an idea of what we have here, we have the correlation. So that's one single patient. Each dot is one trial where we correlate the heartbeat evoked response on the y-axis with a self-rating. So the correlation is not perfect, but it's still a correlation about what someone is thinking about with just one neural index. Another way to look at the self was to see what happened where people imagine either themselves in a given scenario. So here, for instance, yourself petting a tiger, or someone else like your best friend in a space rocket. And we looked at responses to heartbeats while they were imagining, either from first person or third person perspective. And again, we found differences in responses to heartbeats, this time a clear polarity reversal in posterior cingulate cortex, but also in SMA and more premotor regions. So those results indicate that there is a link between the heartbeat evoked response and the first person perspective that you need for conscious perception, or for when the self is explicitly engaged. So the next step was to move to more cognitive aspects where self or consciousness are less obviously engaged. So we turned to decision making, and this is a work from Damiano Azzalini, where he asked people whether they preferred Forrest Gump or Matrix. Okay, so typically in decision making, people think that this is done by you rate those two items, and then you select the one with the highest value. So we could retrieve in MEG the value network that engages in particular the posterior ventromedial prefrontal cortex. And the question we had was whether heartbeat evoked responses would interact with the valuation process. Because when you are asked about your preferences, those preferences say something about yourself, about who you are. So there must be something related to the self for this type of task, which is very different from saying, do you want 10 euros or 5 euros? Where here, there's nothing really self-related. And indeed, there is an interaction between the heartbeat evoked response and the value encoding that is presented in orange in the time course here. And this interaction predicted behavior in the sense that the larger the interaction, the more stable people were in their preferences, but also at the single trial level. So when there was a large interaction, people were more precise in their choice. And when the interaction was smaller, people were less precise in their choice, in the sense they were more inconsistent from one trial to the other, saying sometimes they would prefer Forrest Gump and another time that they would prefer Matrix. So it seemed that there is a link between the stability of preferences that say something about our identity and the interaction between subjective valuation and heartbeat evoked responses. So I'll go briefly through another work that we did together with Diego Candia in a collaboration with Coma Science Group in Liège, which was to see whether we could use heartbeat evoked responses to detect consciousness in patients. So those patients are emerging from coma with disorders of consciousness. So they could be either minimally conscious or in the unresponsive wakening, with the unresponsive wakening syndrome. And we wanted to know whether heartbeat evoked responses could distinguish between those two cases. So we analyzed 68 patients. We had a training set, a test set, and in the test set, we found that indeed heartbeat evoked responses can distinguish quite nicely between minimally conscious and non-conscious patients. How well a heartbeat evoked responses predicted consciousness state actually correlated with the glucose metabolism that was measured simultaneously in two brain regions, in the ventral visual pathway and also in the anterior superior temporal sulcus, which is, again, a node of the default network that seems to pop in pretty often in our data. Okay, so we now have a bunch of evidence relating heartbeat evoked responses to different aspects of consciousness, and we think it's because it addresses this underlying first-person perspective. I just want to use 2-3 minutes to present you some stuff about the stomach because it's fun, but it's less well advanced. So what you probably do not know that the stomach is generating its own electric activity. So it contains a specific type of cells called the interstitial cells of Cajal that are electrical pacemaker. So they are not neurons, but they have electrical properties, and they generate a slow wave. And by slow, I mean really slow, like a cycle is 20 seconds. And this is how it looks. This is a work of Nikolai Volpert, who analyzed recordings of more than 100 participants. So if you place cutaneous electrodes over the abdomen, you can see something like this blue trace, where you have this slow rhythm, which is a gastric cycle, and superimposed on top of it, you see the faster respiratory cycles, and in the inset, you see the heartbeats, actually. And the gastric rhythm has a very clear spectral signature that you can see on the right with a sharp peak around 0.05 hertz. And we wanted to ask very basic questions to begin with, which was, okay, is there a link between this slow rhythm and brain activity? And it turns out that there is. So Ignacio Rebello, using simultaneous electrogastrogram recordings and measuring bold activity with fMRI, showed a delayed connectivity network. So this is what it is illustrated by those sine waves, where in blue you would have the stomach, and then we are searching for voxels that behave like the red curve or the orange curve, meaning that they have a given phase, so they follow the stomach with a specific phase. And there are actually quite a number of brain regions that behave this way, including in particular primary somatosensory cortex, which would have been expected, but also less expected regions, in particular on the medial part of the occipitoparietal regions. And this gastric network seems to contain regions that have body maps. So, of course, body maps for touch in primary and secondary somatosensory cortex, but also in the occipital lobe, a region which is compatible with the extrastriate body area that lights up when you show body parts to participants, or in the cingulate motor regions, when you prepare to act, there is also a somatotopic organization. But we also found with MEG that the alpha rhythm, the amplitude of the alpha rhythm, is actually coupled to the phase of the stomach rhythm in what is called phase amplitude coupling. And this happened in those regions that you see in the insert outlined in green, so along the parieto-occipital sulcus, and in regions where we do find bold gastric coupling. And interestingly, for the gastric bold network, we don't know who is driving what. So we don't know if it's that the brain is following the stomach or the brain entraining the stomach. But for the alpha rhythm, we could show that it's actually an ascending influence, so that the stomach is actually constraining the amplitude of the alpha rhythm. So not the whole variance, but 8% of the variance of the alpha rhythm is due to the phase of the stomach.
Catherine, 1-2 minutes more?
Yeah, that's my concluding slide. So, but I can't see any longer. Okay, never mind. I just have to imagine my slides. So what I've tried to show in this talk is that on the one hand, there is a constant dialogue between the human neocortex and the stomach, and it's actually it actually engages a large number of regions. So in the case of the stomach, we don't have functional correlates yet, but I cannot imagine that this is something we should not have in mind when we are trying to understand how we interact with the environment, because we interact with the external environment, but obviously our brain is interacting a lot more than we expected with the internal environment. And what we've shown mostly with the heartbeat evoked response is that your experiences and your thoughts and your cognition depend on this dialogue, so mostly by using the heartbeat evoked response as an index. And I want to finish by thanking my collaborators and people in my group who did great work, and that I really thank heartfully. And thank you, everyone, for your attention.
Thank you very much, Catherine. Thank you for this wonderful talk. I'm asking people to actually type in their questions, and I will be relaying this to you. But let me start by asking the following. You know, the question of where the self is located has a very long history in human culture and civilization. You know, there were debates between Plato and Aristotle about the heart and the brain. Now, you know, the guts come into the picture. Do you think we're running the danger of trying to be intensively trying to localize the self somewhere? Should we think differently about this relation between different body parts? What's your view on that?
No, that's a tough issue. I mean, I'm certainly convinced that consciousness is generated by the brain. But I think this relates to this thought experiment by philosophers that if you have an isolated brain, is it a conscious being or is it just an isolated brain? And that's not so easy to answer. And I'm not sure that a brain that would be isolated from the rest of the body would still be a self, in the sense that a self refers to an organism. Mm-hmm. So I think we should distinguish between two different things. One is to understand how the brain is generating consciousness on the one hand, and on the other hand, at which level should we define the self? I think it makes sense to define it at the level of the organism. Mm-hmm. But then, of course, you can reduce this organism to the simplest organization that you need to generate a self, and that leads us to all kinds of tough issues.
Yes. I have one question from one of the audience members. Whether data from EEG results that could directly link vagal nerve activity to activity in the GI.
So I missed the beginning of the question. I'm sorry, Manon.
Was there any data from the EEG results that could mm-hmm. directly link vagal nerve activity to activity in the GI?
Yeah, that's a good point. We don't really know because it's actually not so easy to isolate what is related to vagal activity and what is related to sympathetic activity. But also in the case of the heart, at least, there are also many other ways that that information from the heart can be transduced and transmitted to the brain by somatosensory receptors, for instance, seem to be sensitive to the pulse of blood. So isolating one component non-invasively in humans, I don't know how we could do that properly. So here I would rather rely on the anatomical work done, for instance, in the group of Peter Strick, where they very elegantly distinguish what is vagal and what is sympathetic.
Mm-hmm. Lucilla Cardinalis is thanking you for a beautiful talk and stimulating work. Teresa Aurora is also thanking you for an awesome talk. And I think earlier she was asking about your experiment with Mariana, and perhaps it wasn't clear, but correct me if I'm wrong, but you interrupt people at random points and you ask them what they were thinking about.
Yeah.
And they will say whether they're thinking about themselves or about other things. Is that correct?
Yeah.
So in that sense, there's always an interruption of their thinking process.
Absolutely. But we are looking at the neural data during the thinking process. So I mean, you can try by yourself. If you have to stare at something for more than 10 seconds repeatedly, it's just super boring. So people spontaneously started thinking about something else. So what I did not specify is that, of course, people could skip answering if they were not sure what they were thinking about, if they had a blank mind, or they had to use scales to rate their thoughts. So if they were not sure how to use the scales in this particular case, they could also skip. So, and they had they had a real training session beforehand. So I skipped many of the important details, but Mariana was very careful to make sure that participants rated only thoughts that they were clear in their mind and rated them using the scales appropriately. Yeah.
Okay, and I'll take just one last question, which was the penultimate one that was posted. Is there variability in the gastric rhythm between different people, and is it linked to certain pathologies?
Yeah, so definitely. So here we've been analyzing only healthy participants because if we see an irregular rhythm, we don't know if it's because we have bad quality recordings or if we have a patient, essentially. So we've selected participants with absolutely no gastrointestinal disorder. There is a variability between people, but it's still within a fairly narrow range that you find the gastric rhythm, and there is a very minor difference between male and females in terms of frequency. If I remember correctly, it's slightly higher frequency in females.
Okay, thank you very much. I think we'll have to leave it here and move on to the last talk of today. Thank you very much, Catherine.
Thanks, Manos, for the invitation.
And it's my great pleasure to welcome Antonio Damasio. Good morning, Antonio.
Good morning. Can you hear me?
Yes, we can. Let me just say a couple of words. Darren, Antonio is visible to the audience, right? Perfect. So Antonio Damasio, I don't think that he needs an introduction, but we're going to give one anyway. He's the David Dornsfire Chair in Neuroscience at the University of South California. Of course, author of many research papers, many books, recipient of many awards. But more than that, I think what is important is, and I'm sure that I speak on behalf of many of us who are attending the symposium, that one of the reasons, maybe one of the most important reasons why we're all interested in interoception is the work of Antonio Damasio. I still remember the feeling that I had when I was reading The Feeling of What Happens during my PhD many years ago, and how important I found that book for actually orienting me in this new science of embodiment and affect. And of course, after that, I went back and I read Descartes' Error, and then I followed up with all the other books that Antonio has been writing. So it is a really great pleasure and an honor to have you here with us. I know it's very early in the morning in LA, and we're grateful that you agreed to join us. And we look forward to your talk, which will be on the physiology of feelings. And is it okay if you talk for about 20-25 minutes, and then we take some questions from the audience?
That's perfectly okay.
Thank you so much, Antonio.
Good morning to all. I hope that you are all seeing me and hearing me. Good morning from a very cloudy Los Angeles. Cloudy in many ways, but the weather certainly. Of course, I'm very pleased to join you. You are a very special group for me because I know that many of you have interests that are extremely close to mine. And of course, I thank Manos very much for inviting me. So there's this title about the physiology of feeling, and this is something that is sort of never ending. We keep digging and we keep finding more interesting things, and we still do not have all the answers that we need to have. But what I plan to do in this brief talk is discuss a series of new findings and new perspectives from work that we have been doing here at the Institute in Los Angeles. It involves work from many people in our group, but in particular in this case, work from Gilles Carvalho, who has been working with us for several years. So I would just like to start to focus us. Unfortunately, I cannot see you, any of you, and I did not hear what came before. But I would like to start with the definition of feelings. Probably this has been done before, but let's do it anyway. So I wrote it down to make sure I won't fail a word. Feelings are mental experiences of body states related to ongoing homeostatic processes. And I think every word counts in this definition because, first of all, it's the acknowledgement that we are dealing with mental experiences. We're not dealing with actions. We're dealing with something that is mental in the true sense and the classical sense of the term. Second, that it relates to body states. This is, of course, the core idea. And the other is that these body states are related to the homeostatic process. So we are, thanks to what we know of general biology and what we have acquired through our understanding of the evolutionary process of life, we are connecting this to this very, very central process of homeostasis, which, of course, has to inform all the work that all of you and our group is doing. So the most important thing here, tumbling from this definition, is the central problem, and this is a problem that I'm sure has been already discussed today for you, and it's a central problem in biology, in psychology, and in philosophy. So we start with something that is in the body, that is truly physical in the best of senses, and we end up with an experience in mind. So this is a very big pirouette, and we need to understand in many, many aspects of our work how we go from a physical object to a mental process. And this is the classical problem. The typical approach in this specific idea, the idea of the physiology of feeling, is, of course, use a classical standard perceptual process. We begin with a model, with an object, and which are body states, of course, and we attempt to copy, quote unquote, that particular model, and that model ends up being our mental state, which is the feeling state. And this is, of course, it requires quite a lot of magic to do, but it isn't magic. It's just very complex and very true to our biology. And of course, we even have a channel to do this magic, and that's interoception, which is the point of the work of so many of you and of ourselves. And of course, this approach is fine, except that by taking the idea that there is something standard in perception, for example, visual perception, which has definitely been the model for so many people working in this area and in related areas, such as, for example, consciousness studies. The problem is that there is nothing in interoception that resembles the standard canonical visual perceptual process. It is something very different on many counts, and it is something that it is known to be different, and yet, much to my astonishment and probably the astonishment of many of you, there continues to be an acceptance that the processes somehow resemble each other. You all know how one of the most important and influential series of works and thoughts on consciousness actually came to be totally realized on the basis of visual perception, which is something that astonishes me to this day. And I was there to see it from the people who fell into that trap, and I could never understand why people could fall. Anyway, so what I would like to do is say that there are reasons why the physiology of feeling is in fact possible. There are some reasons why some facts that allow us to advance a little bit in our response to this quandary about the transformation of the physical into mental. And there are two things which I wanted to report to you. One has to do with the design, with the frame of this process of interoception, and the other has to do with specific physiological features, which, to my considerable astonishment, have not been, some of them have actually been described for a long time, but have not been exploited for their power. And some of them we have been repeating in experiments in the lab to verify that in fact they exist. So the first has to do with the design and the frame of interoception. And there's something here that is very curious and has to do with the problem of the container and the contained. Obviously, there is a container, which is the organism's body, and that organism's body is in fact the object of interoception, and there is something contained in that same organism, and that is the nervous system, and that the nervous system in itself contains the subject of this process. And this is something that is, of course, very apparent, and yet is constantly glossed over. Then we have I'll return to this, then we have the issue of the physiological features that are particular. Well, of course, everyone knows that the interoceptive system has a very remarkable distinction in relation to others, both in terms of the central nervous system organization and the peripheral, and that has to do with the lack of myelin. And what is interesting here is that this has always been so. We're dealing with a system that is, of course, very old evolutionarily, and that, in fact, never developed myelin. But of course, one could ask the question, why didn't it? So many things have been retired in the course of evolution by natural selection, and have been retired because they were not doing a good job. Why didn't we ever retire the lack of myelin? Why didn't we promote the appearance of myelin in interoceptive systems, which we could have, the same way that it has been gloriously promoted in anything that has to do with general cognition, that has to do with movement, that has to do with language, has to do with the fine perception, for example, in the visual system, which does very well with fully myelinated neurons. So the fact that there is a chronic lack of myelin and that this continues to be is certainly a very strong indication that this lack of myelin is useful functionally and it's delivering something that nature decided not to do without. Now, the lack of myelin, what happens, of course, in the form of denuded axons throughout the system, peripherally and centrally, and it promotes a variety of secondary effects that are very intriguing. One is that it promotes non-synaptic signaling. We have this remarkable fiction that everything in our brains operates with the help of synapses, and this is simply not true, and this is particularly not true in terms of this foundational system, which is our interoception, where probably more than 80% of all neurons are in fact devoid of myelin, and where non-synaptic signaling is galore. We have axons that do not have normally myelinated axons have points like the Ranvier nodes where you have the possibility of interference from the exterior, from the surround. But here the entire axon is a free-for-all. It can be contacted. Not only can it contact, but it can also function as a secretor. So in fact, there are a variety of molecules, including classical neurotransmitters, that are secreted by the axons into the environment, and it's an environment that is not protected, is not insulated by myelin. So in addition to promoting non-synaptic signaling and the secretion of molecules, you have the possibility of being affected by the surrounding molecules. And you have also other interesting phenomena, such as the phenomenon of ephapsis, which has been entirely forgotten, and which is, for example, very, very intense in major channels, such as the vagus nerve, where you have a palisade of parallel denuded axons. And ephapsis is so forgotten that, in fact, you cannot even find it in the list of references in major textbooks of neuroscience. Then there's something that is even more interesting to me, which is the lack of protection from blood-brain barrier, and not only the blood-brain barrier, but the blood-nerve barrier. Now, everyone knows about this lack of blood-brain barrier in certain areas of our central nervous system, like the area postrema and the circumventricular organs, but it so happens that it's there patently in every ganglion that you can think of along the spinal cord and along the spine and the brain stem. So, for example, the nucleus tractus solitarius is the nucleus where there is not protection from blood-brain barrier. Every dorsal spinal ganglion, every point of entry of information from our entire body into the central nervous system at the level of the spinal cord has spinal ganglia, and lo and behold, those spinal ganglia have a cell-rich area, and that cell-rich area is devoid of blood-brain barrier. So what happens is that you have a commingling of signals from the surrounding circulatory apparatus and the cell bodies of those neurons. This is absolutely spectacular. Sorry not to be showing you an image, because the images tell a very, very big story. So you literally have this bunch of cell bodies, and all around you have circulation of blood, and you do not have this protection that myelin has been giving. So I think when you put all this together, you have a possibility of commingling of signals, which is absolutely different from anything that can happen in your auditory system or that can happen in your visual system. So you have this bizarre situation in which you have not a purity of signals from one point to the other, from parts of the body into a central nervous system, but rather an impurity of signals, such that the messages as they are coming into the central nervous system are being influenced by the body in the broad sense of the term, because they're being influenced by changes in metabolism, by all sorts of changes that are happening in the non-neural surround of these channels. But then you have the possibility of the central nervous system responding to those changes directly, and in a very curious way, changing the object of perception. So you have a particular object. That object is being, quote unquote, copied and represented and sent over into central apparatus, and that central apparatus has the possibility of saying, wait a minute, I don't particularly like you as object, I'm going to change it. And this, thankfully, this is actually what happens to us in pain conditions in which the central nervous system decides to relieve us of part of the pain even before we take the drugs that would mitigate it. And so this situation is, of course, so dramatically different from what happens when I look outside. I'm looking at the Santa Monica Mountains, and there's no way I can interfere with the structure of the Santa Monica Mountains from my point of view as subject of that particular object, which is the Santa Monica Mountains coming through my window. So all of this is what I really wanted to call your attention to. Sorry about not having pictures to show you, but the pictures are very dramatic. I recommend everyone to look at vagal nerve fibers and see the difference between a myelinated and a non-myelinated fiber. And I think that this gives us an entry into the classic problem of how you transform the physicality of a body into the mental experience of a feeling. We are not dealing in any way with classical perceptual processes. We're dealing with a mixture that begins with the get-go. It's a mixture that begins with object and soft. There's a complete commingling, and that is what will give us some chance of placing this in the perspective of consciousness, for example. Let me just close by saying I do not believe for a second that we can deal with the so-called hard problem of consciousness without dealing with the problem of feeling. The problem of the hard problem of consciousness is hard if we're trying to deal with the problem as if it were a classical perceptual problem on the canonical visual perception model, but it is not. It is totally hooked with the process of feeling, which is a fundamental process of projection of homeostasis, and which is the process that allows us to be informed about body states, good, bad, or indifferent, and gives us the incentive to do something or do nothing about those body states. If they're good and convenient for our life, let them be. And if they're not good, let's try to do something about them. And that is the patent function, quote unquote, of the feeling systems. And those feeling systems begin with interoception. They don't begin with other types, eventually much more perfect and evolved of our perceptual systems. And that's where we are. Thank you very much, and Manus, to you.
Thank you very much. Thank you very much, Antonio. And I'm sure everyone will be clapping in their living rooms or offices. Thought-provoking as always, and we do have some questions from the audience, and I will be relaying this to you. The first one is from Harry Farmer, who's thanking you for your talk. And he's wondering, what implications do you think that the free-for-all nature of the unmyelinated interoceptive signals has in terms of using computational models to probe interoception?
That's a beautiful and deep question. I think it requires that we do not use the computational models that have been perfectly designed according to classical models of neural functioning. So if we stay with classical zeros and ones and with classical synaptic signaling, we're not going to go very far. We have to begin by acknowledging that these phenomena are different. This system operates according to different rules, and we need to incorporate that in the modeling. Does it make it more difficult? You bet. Much, much more difficult. But at least it will be real, which is good, as opposed to a fiction.
Yes. Next question from Patrick Haggard. He's thanking you for the inspiring talk. And Patrick says that myelin enhances neural conduction velocity. So one sometimes thinks of interoception as slow, but that is not your interpretation, Antonio, of the significance of myelin. Can you comment on the temporal aspect of interoception? Is it a mistake to think of it as a slow background?
That's also a fascinating question. Thank you. Well, I think it is slow. It's definitely slow. One thing that we have gained with myelin, and the gain is obvious in the motor system, but it's also very, very apparent in auditory and visual processes. You gain speed. You really conserve the signals and you speed them up, precisely because you don't have all that interference along the path. So I don't think it's wrong to think of myelin as slow, of interoceptive operations as slow because of the lack of myelin. I think there is a gain to be had by that slowness, and that actually allows us. Of course, there was no design like that in nature, but in the end, it's something that we can capitalize on by giving us more time to react. And I think when you get messages that are signaling something being wrong with your physiology, and that message is giving you an impetus for response, it's actually good that you have a few more microseconds to manage the response. And also because it allows it to capture our attention and concentrate our mind on that particular process. So I don't see that as, I see increasing velocity a definite advantage, because if we didn't have it in the motor system, it would be a disaster. But I don't see it as a problem for homeostasis. And again, thank you for noticing the fact that I'm tired of this classical idea that, well, interoception is so old. This is so, such, we should have retired. But the real fact that we need to concentrate on is that why wasn't it retired? It would have been so easy, with at least 500 million years of nervous system evolution. It would have been easy to kick this thing out if it were not doing anything very, very favorable to the living organism.
Well, it certainly hasn't retired, and it's certainly one of the topics that attracts a lot of interest, as you know. And your work was pioneering in that respect. I know that there are a lot of young, early career researchers listening to today's symposium, and I wanted to ask you, what do you think are the important challenges that the new scientists should try to tackle in this field of research in interoception? What do you think are the most important things that we haven't done already, things that we should be trying to look into more carefully?
Oh, well, how about everything? No, I think it is very challenging. I think the first thing that we need to do, and that's what you're doing so well, is calling attention to the problem. Because it really is astonishing to me how little this has been considered. Some of the findings, I asked a number of colleagues that I very much respect in neuroscience, that I consider giants of the field. For example, about the lack of blood-brain barrier in spinal ganglia. I did not find one person that had heard of it. And these things have been described in the literature. We were trying to do it a little bit better and investigate a little deeper the problems, but the literature exists. And some of it is not from last year or the past few months. So it's a question of the optics being completely different. I'm old enough to remember when the Society for Neuroscience started, and there was not even a session that had to do with affect, nothing that had to do with emotion, which was at the time how we would describe our entire field, unfortunately. Yes. But it was all absent. It took until 1995 to have the first session of the Society for Neuroscience about emotion. So that tells you. So I think that the main thing we have to do is to send out the word. And fortunately, as you have from this group, you can see that there are lots of people that are producing extremely interesting work. And this has to be heard. And eventually, if we have science foundations that will help work post-COVID, then we get to work and do it.
Yes. And I think you said it very nicely, and you explained what is the physiological issues that we have to confront when we study interoception, this impurity and the messiness of it all, because that's what the visceral body is all about. And at the same time, interoception is a very difficult system to actually manipulate in an experimental way. It's not like vision. You can very easily control what you actually present to your participants. And this difficulty in getting a good experimental control over it, the impurity of it creates a lot of methodological challenges. And of course, as scientists, we want to find the pure pathway from the body to a mental process, but maybe that's not the case with interoception, and we have to accept the fact that it's going to be a bit messy, and we're dealing with a system that has this inherent impurity.
Right, and deal with the messiness as part of the advantage, because the messiness is exactly what is part of the messiness has been very important, and that's why we have kept it like that, I suspect. Yeah, and certainly allows us to do a lot of beautiful things. in terms of the control of our behavior and in terms of how we project ourselves into the future.
Thank you. I think what is the important take-home message is that we should actually consider this impurity and messiness as an advantage for driving the field forward.
Absolutely.
I would like to thank all of the people who attended our symposium today, all the speakers, Micah Allen, Sarah Garfinkel, Katrin Talon-Baudry, and Antonio Damasio. For me, it was a great opportunity to bring all of you together. It would have been very difficult to do it in a physical location. So one of the few silver linings of the current pandemic is that we have this opportunity to create things like that in the online world. Under normal circumstances, I would be taking you out for drinks. Unfortunately, we cannot do that, and it's very early in the morning in LA. But thank you all for tuning in. I know that the talks will be available online later on. It's been a great pleasure chairing the symposium. Thank you to all the speakers, and have a nice evening and a good morning to you, Antonio. Thank you very much.